Imagine a world where illnesses like cancer and sickle cell can be cured or vital organs can be repaired. What ethical dilemmas will CRISPR present to social and political platforms? How would it affect our communities, and what would the world look like?
As Socrates states, the unexamined life is not worth living. We are curious beings by nature, and that curiosity has led us far. All throughout history, we have been curious about what makes us human, from our way of thinking, our intelligence, to our human physiology. Yet with such in-depth exploration of oneself, there are external societal consequences. How far is too far in changing our human composition and the environment around us?
The discovery of the double helix structure of DNA in the 1950s revolutionized the understanding of DNA. Out of that discovery came the development of gene editing technologies behind gene-based medicine and therapy. Since then, incredible new gene-based technologies have been developed, one of them being clustered, regularly interspaced short palindromic repeats, or in short, CRISPR. This fairly new technology allows for scientists to selectively edit the DNA of living organisms. Who first discovered CRISPR has been debated, but a consensus has been made that it was first discovered by Japanese scientists in 1987. Multiple labs have been working on CRISPR for years. However, a Spanish scientist, Francis Mojica, coined the name, which later led to an eventual CRISPR-Cas9 breakthrough. Credit for the discovery has been given to Jennifer Doudna and Emmanuel Charpentier in 2012, but other teams contributed important work and thought as well.
To better comprehend CRISPR, it is important to understand a few fundamental concepts. What is DNA and RNA? What is gene editing and how does it work? What is CRISPR versus CRISPR-Cas9?
According to the National Human Genome Research Institute, DNA is a molecule that carries genetic information for the development and functioning of an organism. DNA is made of two linked strands that wind around each other to resemble a twisted ladder, a shape known as a double helix. In other words, DNA determines how we will develop and look physically. RNA is a nucleic acid present in all living cells that has structural similarities to DNA. Unlike DNA, however, RNA is most often single-stranded. Essentially, RNA carries the instructions of how to produce proteins and allows for DNA to be read in a living organism.
Gene editing seeks to modify the genes and make specific changes to the DNA of a living organism. These modifications can be made in both non-heritable and heritable genes. Gene editing technologies are able to add, remove, or edit DNA. So, what is CRISPR?
Many tend to use CRISPR and CRISPR-Cas9 interchangeably. CRISPR is a sequence of DNA found in bacteria and archaea. At its inception, the role of CRISPR was to detect infected DNA sequences and destroy them as an antiviral system. CRISPR-Cas9 works by correcting, cutting, or editing out DNA and letting the natural DNA repair process using Cas9 enzyme and mRNA take place. This revolutionary gene editing tool aims to completely change gene-based medicine and therapies. However, it is important to take into account how it will change global health and its ethical implications. CRISPR is paving the way in the very conceptualization of our health as a species. The list of conditions CRISPR can treat is long, but two of the most exciting potentials are the treatment of blood disorders and cancer.
Think of that. Tools to prevent cancer have been spoken of for decades, but now they are on the cusp of reality. CRISPR can one day do this by editing immune system cells, engineering them to attack cancerous growths. Speaking of disease, for example, CRISPR can also fight dangerous infections. It can identify and diagnose threatening pathogens in an efficient, timely way. This means that CRISPR could be a solution to viruses and bacteria that have plagued humans for generations, such as malaria. Of course, when there is an exciting new innovation, there is always misinformation surrounding it. First of all, there is the idea that CRISPR will bring about superhuman designer babies. Although this has a grain of truth, reality is often disappointing.
These traits are controlled by many genes, and trying to pinpoint which genes we must edit to change one's intelligence is an impossible task for the current time. There is also confusion about the difference between CRISPR and GMO when it comes to crop modification. GMO and CRISPR are not the same process. CRISPR allows for a very specific edit to be made in a crop, while traditional GMO technology introduces a foreign body into the crop.
For any lofty ambitions, there are, of course, challenges and limitations. CRISPR is no exception. One challenge is a question of accuracy. Cancer is already a mutation of the human body, and CRISPR, if used without caution, could cause off-target effects, that is, unintended mutations of the genome. Even more, CRISPR could accidentally increase pathogens' propensity to mutate, stimulating its desire to survive.
There are technological externalities hindering CRISPR's development as well. Methods to deliver CRISPR into DNA must become advanced enough to prevent unwanted outcomes and limit immune response. On top of this, technology related to the accuracy and precision of CRISPR treatment must be further developed, as well as other technologies in the biomedical field, such as microscopes, cell culturing equipment, and analysis machinery. When it comes to gene editing, there are worldwide ethical considerations as well. Who receives CRISPR treatment? This technology must be promoted with equity. Increasing wealth inequality and stressed international relationships will make this very tough. Another contested debate is the potential of eliminating physical and mental disabilities. This forces nature to create a normative, brave new world. A world in which differences may no longer be celebrated, but abhorred.
With all of this said, CRISPR is still in its early development. However, it offers a promising future. A future of treatable cancers, regenerative organ transplants, treatment of sickle cell, diabetes, and potentially hereditary mutations. Perhaps even one day, advanced embryo editing can become reality, offering intelligence that can be measured before birth. Technologies like CRISPR are in continuous development and will have significant effects on the future of our society. How will this future affect you?